Hello students, welcome to Tungala online classes. So in the last video we were discussing the functions of the cell membrane or the plasma membrane. Many more functions it does itself, you came to know of course. And after that one I had started with the cell wall, and this cell wall is an outermost rigid very hard structure that encovers the cell and which gives the particular shape for one particular cell, right? And chemically what it is made up of already we have gone through. This cell wall not only present in the plant cells but also present in some other organisms like prokaryotes. You know it better. So prokaryotes like bacteria, cell wall is present and it is made up of peptidoglycan. Thoroughly we have studied. So there glycocalyx. So all these are the things that we have studied in case of bacteria, that is prokaryotic cell. There also cell wall is present. And after that one, fungi, they have also some of the fungi. So have the cell wall which is made up of chitin. Chitinous cell wall is present in case of fungi. And along with this one, some other cryptogams like algae. Algae have cell wall which is made up of cellulose, galactose, mannose, silica, calcium carbonate. So this is very important point to be noted over here. Algae cell wall is made up of cellulose, galactose, mannose, silica, calcium carbonate, etc. Right? So all these are the chemical components of all other organisms, so which have cell wall, right? So now we are highlighting the cell wall of the higher plants itself, so which is made up of cellulose as well as pectin, other materials, right? So what are those chemical compositions that we have already studied in the last video? Now in this video thoroughly we will start with the structure of the cell wall now itself, right? Let us start with structure of cell wall. In case of plants. So how the cell wall structure is? The cell wall structure, it has three major sections already I have mentioned. One is called as middle lamella, second one is called as primary wall, and third one is called as secondary wall. And rarely in case of lower organisms, lower plants like gymnosperms, tertiary wall is also present over there. So let us study one by one thoroughly now. So let us list out the parts of the cell wall. Cell wall has the following sections. What are those? Middle lamella. Middle lamella is one. Primary wall. Second one, secondary wall. And fourth one, tertiary wall. Very rare. Right? So all these are the major sections of the parts of this cell wall of the plants, right? So let us see here, middle lamella is the first one. So let us start with the middle lamella, right? Middle lamella. What is this middle lamella? It is the cementing layer that lies commonly between two adjacent cells, right? So it is a cementing rigid wall present between two adjacent cells. It is common for both the cells and that is called as middle lamella. Diagrammatically, let us see now itself, right? So if this is one plant cell which is hexagonal in nature, right? We'll see, this is one plant cell, and yes, this is the plant cell what we are seeing on the board now. So it has a common middle lamella. This is said to be as middle lamella, right? And inner to the middle lamella there is another wall which is called as primary wall is present over here, and inner to the primary wall there the secondary walls that we come across, even the tertiary wall that we come across over here, right? So but what we are studying here is middle lamella. First of all we'll start with the middle lamella. What is this middle lamella? It is a common cementing layer lies between two adjacent cells. Between two adjacent cells, it is commonly lying over here. That kind of a cementing wall itself is called as what? So the middle lamella itself, right? After this middle lamella, inner to this there is one more layer that is called as primary wall. So we'll see it later. So chemical composition. When we go to the chemical composition of this middle lamella, there we come across what? It is made up of calcium and magnesium pectate. It is made up of calcium, magnesium pectate, and the formation of this middle lamella starts during the end part of the telophase, right? The formation starts. So the formation of this middle lamella starts during telophase, end of the telophase. Especially we can consider during cytokinesis. During the cytokinesis, so what happens over here? So at the end of the telophase, so which is the beginning of the cytokinesis, that moment what happens? The formation of this middle lamella starts, right? It starts over there, right? See here, if this is the plant cell, then this plant cell has the nucleus over here. Its division starts over here, and later here the cell plate is formed over here. What is formed? Cell plate is formed, and this cell plate formation starts at the end of the telophase, and during the cytokinesis it starts. Okay, so this does not reach the end of this wall of this cell, right? So initially this starts during telophase. That moment what happens? Accumulation of the fragments of the endoplasmic reticulum, as well as here accumulation of fragments of endoplasmic reticulum, as well as Golgi complex that takes place over here. So what are the major parts of Golgi complex as well as endoplasmic reticulum that forms the wall over here due to their accumulation? So later I will explain. So whenever we are studying endoplasmic reticulum as well as the Golgi complex there, alright? The fragments of endoplasmic reticulum as well as Golgi complex, they start to accumulate over here and form a cell wall or the cell plate. It is called as what it is known as cell plate. And this cell plate is formed during cytokinesis. So that what happens when this cell plate that completely reaches the end, at that moment this becomes an independent cell and this becomes an independent cell. So now I hope you are so clear that due to the accumulation of the fragments of endoplasmic reticulum as well as Golgi complex, so forming the cell plate during the cytokinesis, this middle lamella is formed. This becomes the middle lamella there, right? So this is the fact what you should know, right? So that point to be noted over here itself. Then what happens? So this cell is adjacent cell. This cell is also adjacent to this cell. So during the separation of the cells, so how these two cells that separate, so when they separate is, so whenever the dissolution of this middle lamella that take place, when middle lamella dissolves, that moment two cells are separated. Now both the cells are jointed because of the presence of a common cementing layer called as what? This middle lamella, right? Here I shall write two cells separate when middle lamella dissolves. When middle lamella that dissolves. When it dissolves, that moment what happens? Both the cells are separated over here, right? After the dissolution of this middle lamella, what happens? So when it dissolves, is the thing what you should know. See here, whenever it dissolves, so the cells become ripened. The fruits, when they ripen themselves, that moment that middle lamella has been dissolved. During ripening of the fruits, middle lamella that dissolves. This is the fact what I should know. Right? That point to be noted over here. This is almost the explanation about this middle lamella. It's a common cementing layer which is made up of calcium, magnesium packed itself, and it is formed during the end of the telophase and during cytokinesis. So it helps in formation of the cell plate due to the accumulation of the fragments of endoplasmic reticulum as well as Golgi complex. These are the facts what you should know regarding the middle lamella. This is one cell wall, means common cell wall, and later the primary wall that we start now. Let us start with the primary wall now, right? Primary wall is so lying inside the middle lamella. Now you are observing in the diagram. So this is middle lamella. Inner to this middle lamella there lies the primary wall, and this wall is elastic and extensible in growing cells, right? So note on the point, primary wall, it is the wall lying inside the middle lamella, and it is elastic in nature as well as extensible in nature in growing cells. And such kind of primary wall is so formed as a result of the orientation or the deposition of the wall material more and more. So one on another. So that way the deposition of wall material that take place over here, so that is nothing but cellulose. So when it is deposited, that moment the wall is formed inner to the middle lamella. That wall itself becomes the primary wall itself, right? See here, the cellulose materials or the filling materials that is deposited this way. So when it is deposited that way, so that moment what happens over here? So the one wall is formed inside the middle lamella, and that wall becomes what? Primary wall itself, right? So let us write the same thing now. The primary wall is growing due to addition of wall material, right, one above the other. So this method of deposition of the wall material one above the other, forming one thick wall, that is nothing but primary wall, is nothing but, so it is called as interception. The growth is called as interception. See here, this method of growth is said to be as interception. It is very important point to be noted over here. Interception. This is the method of growing of this wall. Wall grows as a result of the deposition of the wall material. That is nothing but cellulose, right? So it leads to interception. The method of growth itself is said as interception. And chemical composition. Chemical composition of this primary wall. What this primary wall is basically made up of? It is basically made up of maximum hemicellulose. 50% of the hemicellulose is present. And next, so minimum of cellulose itself. And next one is some of the pectic material is present. And along with this one, some proteins are present. Along with this one, non-cellulosic polysaccharides are also present over here. All these together form the primary wall itself. Then what is chemical composition of this primary wall? Let us see here. So here hemicellulose, maximum is hemicellulose, so which is about 50%, and cellulose is also present, which is about 25%, and next one is a little about pectin material. So that is pectin material, which is a little bit along with this one. So some of the proteins are also present over here. Along with this one, some of the non-cellulosic polysaccharides. All these together form the chemical nature of this primary wall. As a result of all these materials, it is flexible in nature as well as elastic in nature in growing cells. Why? Because growing cells stretch themselves. During elongation of the cell, they stretch themselves. That's why deposition of the wall material that leads to interception method of the growth of this primary wall that take place over here. And this primary wall, what it is basically made up of? So whenever we see the structure of this primary wall, that moment, so it consisting of their elongated fiber like structures called as macrofibrils. So primary wall is made up of fibers like structures called as what? They are called as macrofibrils. Imagine yourself, so this wall is made up of basically the macrofibrils. I shall draw here the diagram. This is one macrofibril you consider, right? So this macrofibril, what it consisting of? It consists of many number of microfibrils. Means this macrofibril consisting of many number of the microfibrils, inside which microfibrils are present. So one of the microfibrils I am going to highlight over here. See here, this is microfibril. So that I am going to highlight over here. This is one microfibril I have removed from this macrofibril. Okay. So these microfibrils are parallelly arranged inside the macrofibril. So they are parallelly arranged inside the macrofibril, and that microfibrils, they are about 250 in number in each macrofibril. So one macrofibril has 250 number of parallelly arranged microfibrils are present over here. Is it very clear? Now, this is only one microfibril I have highlighted over here. This consisting of what? This also consisting of many number of fibrillar structures. They are so mycellae. Each microfibril that contains mycellae, right? Mycellae are present in each microfibril. So these mycellae are about 20 number in each microfibril, right? So this is also fibrillar structure. And imagine yourself inside this mycellae, there hundreds of that is 100 number of what are present inside it. What are present inside it? They are nothing but cellulosic chains. 100 number of cellulosic chains are present over here in each mycellae. So this is how the wall material is basically made up of fibrillar structure called as macrofibril, which contains parallelly arranged 250 number of microfibrils, and each microfibril consisting of 20 number of parallelly arranged mycellae, and each mycellae consisting of 100 number of cellulosic chains, and each unit of the cellulose itself is glucose, right? Each unit of cellulose itself is glucose. Chemically, cellulose is made up of glucose, right? So this is how the wall material that is deposited so as to form the primary wall. Of course, imagine yourself, there are some macrofibrils are there. They are arranged on this middle lamella so as to form the primary wall. And such kind of macrofibrils contain microfibrils contains mycellae contains the cellulosic chains. This is the fact. Apart from these macrofibrils which are arranged parallelly, so what is other thing apart from macrofibrils? Right, so inside which what is present over here? Matrix is present over here, or the ground substance is present over here. And what is this matrix mainly consisting of? So what is this matrix mainly consisting of? It is also consisting of chemically the water as well as hemicellulose as well as the pectin materials are present over here along with some glycoproteins. What is this matrix mainly consisting of? It consisting of water. It consisting of hemicellulose and it also consisting of the pectin material, and it also consisting of glycoproteins. All these are present over here. In that, pectin act as means in this matrix matrix. So the microfibrils are lying over there, and this pectin material in ground substance, it helps as the filling substance. The gaps are filled with the help of this pectin material. So it acts as the filler materials over here. It acts as the filler material. So what about this hemicellulose? So it binds the microfibrils in matrix itself, right? It binds microfibrils with matrix. With matrix it binds, right? So that is the nature of what? Hemicellulose, right? And what about these glycoproteins? Glycoproteins are also present over there. Water is also present over there. All other materials are present over here as a ground substance inside it. And so the orientation of the fibrils that is taken place over here due to all these ingredients, right? So this is what the primary wall. What is the major function of this primary wall? So the major function of this primary wall is, so it helps in giving the strength, it gives rigidity, right, and protection to the cell or the protoplast itself. It gives strength, rigidity, as well as protection to what? Protection to the protoplast. Protoplast is nothing but, so inside the wall there is a membrane. What is that? Plasma membrane is there along with the cytoplasm, all other ingredients of the cytoplasm. So all together called as what? Protoplast. To that protoplast it gives rigid nature, strong strength itself. So that is given by this primary wall. And one thing you should be very clear that, so the meristematic cells have only the primary wall in them. Meristematic cells or meristans have only the primary wall within themselves, right? So this point to be noted over here. So this is about what? The primary wall. And next one is the secondary wall. Let us start with the secondary wall. Secondary wall, right? And this secondary wall which lies inside the primary wall. So this secondary wall which lies inside the primary wall. This is 1, this is 2, and this is 3. What it suggests us is, so this secondary wall is 3-layered one, right? So this secondary wall, it is thick, rigid wall, lies inside the primary wall, right? And this secondary wall is of 3-layered one, right? Secondary wall is 3-layered one, and they are named as S1, S2 and S3. S1 secondary wall 1, secondary wall 2, secondary wall 3. So like this there are 3 walls which are lying inside the primary wall. It is the highest thickened wall in plant cell. Remember this point to be so noted also, right? So secondary wall is also made up of means chemically it is made up of cellulose, so which is deposited, right? So which is deposited one above the other, means these so layers which are arranged one above the other, and this kind of growth of the wall itself is called as accretion. What it is known as accretion. This point to be noted over here. Accretion is nothing but the deposition of wall material take place one above the other so as to form the secondary wall, right? So this is more important point to be noted. Accretion is very important word to be remembered, right? Next one. This cellulose, not only the cellulose, the deposition of other materials also take place in the secondary wall, right? So and one more point is, this secondary wall is formed after the enlargement of the cell. The point is very important to be remembered. So what is that? This secondary wall is formed after the enlargement of the cell itself. After the complete enlargement of the cell only, the secondary wall is formed over here. So especially in some of the plants like Euphorbia milli is the best example for this one, which secondary walls are seen over here. So these walls are formed as a result of the deposition of the material one above the other. That kind of growth of the wall itself is called as accretion. So very important things to be remembered over here. So and come to the next point. So this wall is also deposited with other materials like, what kind of materials are there? Pectin, hemicellulose, cellulose, all these are the other materials which are deposited over here in the secondary wall. Let us write down the wall materials like pectin is also deposited over here, and hemicellulose is also deposited over here. Suberin is cutin, some of the waxes. So all these are deposited over here on the secondary wall. See here, pectin, hemicellulose, suberin, cutin, waxes, as well as the lignin is also there. Lignin material is also deposited over here, right? All these are other materials which are deposited on the secondary wall, and this secondary wall is formed after the complete enlargement of the cell only, right? So and the material like lignin, which is deposited so as to form the secondary wall, and that process itself is called as the lignification. What it is known as lignification. What do you mean by lignification? The process of formation or the process of deposition of lignin material on the secondary wall. Deposition of lignin material on secondary wall. On secondary wall is called as what? It is called as lignification. It is called as lignification, right? So this is more important point to be noted over here. And one more thing is suberin is also there, right? Suberin usually it is deposited in the endodermal cells, as we have studied in the anatomy. So endodermal cells are deposited with what? The suberin material, and even the cork cells are also suberized over here, and the cork cells as well as endodermal cells both are deposited with what? Both are deposited with suberin. Both are deposited with submarine material, and so this submarine material itself is so impermeable to water as well as gases itself. As we have seen in anatomy, so there we came to know during the absorption of water, only the water passes not through the endodermal cells, only the limited endodermal cells, they are called as passive cells. All others are impermeable in nature. So for water as well as gases, already have told you. Right? This is one fact what you should know regarding the deposition of submarine in some of the endodermal cells as well as cork cells. You don't know about the cork cells, so they are also a kind of a dead cells. There we come across. Okay, so next one, the orientation of these macrofibrils that take place in every cell layer. I mean every cell layer of the secondary wall. So it is different. Orientation or the arrangement of the macrofibrils in every layer is different. Right? Point to be noted. See here, if this is the secondary wall, if they are arranged like this, so in this one they are arranged like this. So in this one they are arranged like this. That differs from one layer to another layer. One more point is, in different types of tissues also, the secondary wall in which the arrangement of these macrofibrils is different for different tissues even. Parenchyma tissue, it is different. Collenchyma tissue, it is different. So sclerenchyma tissue, it is different. Like this, in different tissues also the arrangement or the orientation of these macrofibrils in the secondary wall is totally different in different tissues. So that point is also to be remembered over here. So this is about the secondary wall in some of the plants. Come to the tertiary wall. What is this tertiary wall? Where actually it is present? It is present in the tracheids of gymnosperms. As you know, water conduction that take place through the tracheids in case of gymnosperms, and those tracheids, the wall is deposited with some materials, so that forms the tertiary wall, so which is very rare in all other plants, right? So, and the tertiary wall is lying inside the secondary wall. Okay, so let us write here tertiary wall. It is lying inside the secondary wall itself. Tertiary wall is lying inside the secondary wall, and it is deposited with, or it is made up of xylem. It is made up of chemically xylem material, and this tertiary wall is present in tracheids of gymnosperms. Tracheids of gymnosperms, they are present in. So that is about the tertiary wall. So this is all about all the wall layers that we come across in the cell wall of the plant cell. So plant cell has the middle lamella, primary wall as well as secondary wall, as well as the tertiary wall very rarely in some of the plants like gymnosperms. Okay. Next one is pits as well as plasmodesmata. These two are very important things. What are these pits, where these are present? Let us see now itself. So I shall draw the diagram of the pit over here. So this is a common wall which is a cementing layer between the adjacent cells. It is called as middle lamella. It is middle lamella. To this middle lamella, there we come across the primary wall is present over here. Primary wall I am showing you here, or else you now to show this one. This is primary wall. On this primary wall you will come across the secondary wall over here, and that secondary wall has 3 layers: S1, S2, S3, right? So this is S1, S2, S3. So these are the 3 layers that we come across, and this is nothing but the secondary wall. It is secondary wall. Okay, so now you came to know how actually the middle lamella is there, and the arrangement of the primary wall on this one, and on the primary wall there you come across secondary wall. So this secondary wall is also present on this side also. You can show this side also. This is the secondary wall: S1, S2, S3. So like this you will come across over here. This is the wall what we are showing here in the case of plants. Now, so let us start with the pits. What these pits are? So what these pits are? You are observing here there is no deposition of a secondary wall. There is no deposition of a secondary wall in certain points or certain areas, and those makes the small depressions in the wall, and those small depressions in the secondary wall are called as pits. Is it very clear? So in some areas of secondary wall there is no deposition of wall, that is secondary wall, and it looks to be as a depression, and that depression is called as pit. That depression is known as pit. Okay, we came to know what is pit. Now you are observing here in the secondary wall, here is one pit and here is also one pit. So the pits of the adjacent cells are opposite to each other, and this is said to be as pit pairs, right? It is said to be as pit pairs, right? So what are pit pairs? So two pits of neighboring cells are lying opposite to each other. It is said to be as pit pair, and the cavity what we come across, or the depression what we come across, is called as pit cavity. What do you come across? Pit cavity. You will come across pit cavity. There is a depression of the cavity that you come across. It is called as pit cavity. Pit pairs means what? So you came to know. And pit cavity means what? You came to know. Okay. So, and one more thing is pit membrane. One more thing is pit membrane. What is this pit membrane, you may ask for here. See here, this is the primary wall, this is middle lamella. So these two together form the pit membrane, right? Pit membrane, so is nothing but, so here middle lamella as well as the primary wall together form the pit membrane. And it is porous in nature, which allows many materials to pass through, right? So this is the fact what you should know about these pits. There are different types of pits that we come across. One is So pits types. One is simple pits. Simple pits. What the pit? What you are observing here? So the diameter of this upper part of the pit and the lower part of the pit almost the same. So such kind of pits are said to be as simple pits. And bordered pits are also there. So I shall show you here. So what kind of bordered pits that you come across over here? So if this is the pit membrane, in that pit membrane what you come across here is pit, right? This is one kind of pit that you come across, which is said to be as the bordered pit. See here, this is secondary wall and this is pit, right? So pit portion, right? Here the secondary wall is projects forward. So when it projects forward, so the cavity becomes larger and such kind of boundary like structure what you have in pits is called as bordered pit. This is bordered pit and this is simple pit. Such kind of many pits that you come across over here, right? So and after this one, next concept is plasmodesmata. Plasmodesmatas. What are these plasmodesmatas? Let us see. Plasmodesmatas are nothing but they are the cytoplasmic connections between the two adjacent cells. So these are nothing but these are cytoplasmic or protoplasmic connections between two adjacent cells is called plasmodesmata, right? So there are two cells. If two cells are there, in between these two cells, so there the cytoplasmic connections what we come across, and this cytoplasmic connection itself is called as plasmodesmata. So this would help for the exchange of the materials between the neighboring cells, right? Neighboring cells exchange the materials, even send or transmit the signals into the cells from one cell to another cell, right? Totally it brings about the linkage between the adjacent cells, right? So this would help in the exchange of materials between the adjacent cells. And so one more thing you must know regarding these plasmodesmata. See here, this is one cell, this is one cell. So there you come across the endoplasmic reticulum in this cell, endoplasmic reticulum is there, and endoplasmic reticulum is also here. The endoplasmic reticulum of this cell that extends into this cell, then it is said to be as what? So that is a kind of connection between two cells, right? And that takes place through desmotubules, right? Here endoplasmic reticulum of one cell, right, connects the endoplasmic reticulum of adjacent cell through a membrane called as desmotubules. This point is very important point for NEET examination. Okay, desmotubules. So right, so this is the fact what you should know. This is regarding what pits as well as plasmodesmata. So this is all about the cell wall of the plant cell, right? Next one is functions of plant cell. We'll discuss in the next video. Thank you.